my curious little champions, I welcome you all to solve top 30 questions from the chapter Microbes in Human Welfare. Well, this is the 10th video of this particular series and I hope this is really helpful for you. Now, there is only one condition before I proceed with the chapter, and that is before I show you the question, okay, and before I show you the answer, not before I show you the question, before I show you the answer, you have to solve the question by yourself. That should be the deal. Okay, no cheating up over here. You should be analyzing your preparation, then only you should be going ahead with the answer that I have. Okay, so if you're not doing that, then you are messing it up. So please not mess it up, children. Please be honest to yourself. Let's begin solving the top 30 questions. The first question. Roquefort cheese is ripened by. So there's a cheese which is known as Roquefort cheese, kind of very costly. Now this is ripened by. It becomes tangy and everything. The whole process of cheese ripening and everything is done with the help of what? A yeast, bacterium, fungi or a virus. Yeast, bacterium, fungi or a virus. Well, this is a direct question that is given from your NCERT. Every question is. But I'm telling you, this is a very direct question. You don't have to use too much of your brain. It's a fungi which is responsible for this. Okay, it is a fungi which is responsible for this. Now, yeast also comes under fungus category, but it is not the one that is responsible for ripening of the Roquefort cheese. Okay, so it is just a fungus. The name of the fungus is not given in your NCERT textbook, but it is Penicillium something else. Okay, there are different. Don't think the penicillin that gives penicillin. Okay, there are other version strains of it as well. Okay, so it is one fungi which is responsible for ripening. Cool. So this becomes your answer. Next, the toxic substance hemozoin responsible for high fever and chill is released in which of the following diseases? So hemozoin is being released in our human body itself. Okay, it is being released in our human body itself. So what happens is that basically when the malarial parasite, okay, when the malarial parasite Plasmodium, one of its stages is basically residing in our RBC, red blood cells. It is feeding on the hemoglobin, and when it feeds on hemoglobin, it breaks them down into heme and globin. But this heme that is there is kind of toxic for this particular guy. So it changes into something else which is known as hemozoin, which actually is not that great for us human beings. So it tried to save itself, but it is going to cause trouble to human beings. And whenever this hemozoin is being released, whenever an RBC ruptures, okay, whenever an RBC ruptures because of this guy generally goes on increasing that number in our body, okay, in an RBC, it ruptures. When it ruptures, it also gives out hemozoin which was there. Now this hemozoin is responsible for what? Fever and chill. So this is associated to which disease, my dear children? It is associated to malaria, not typhoid, not dengue, not pneumonia, it's malaria, okay. Please remember that. And Plasmodium has lots and lots of stages: gametocyte stage, sporozoite stage, this stage, that trophozoite stage. So many stages are there. I'm not going to tell you which stage is the one that is going to infect RBC. No. As of now, you don't need to know that. You need to know who is the one, in which disease is hemozoin actually being released. So I gave you an extra information over here, and I think that's enough. Why I'm not saying is that, not because I don't want to teach you. I have taught you in different chapters. It's because I want you to remember a specific detail over here. Hemozoin, malaria. Hemozoin, malaria. That's all. That's all you need to remember. Otherwise, you'll get confused. Okay? So this becomes the answer, my dear children. Now next question, which vitamin is increased by LAB in curd? What do you mean by LAB? LABA. It is what? Lactic acid bacteria, my dear children. And lactic acid bacteria, I'll tell you, there are a group of bacteria and things like that who have the enzyme responsible for making of vitamin B12. It is what? Vitamin B12, which is kind of really important for us. So in the curd, we say you should be having curd, you should be having curd. Why you should be having curd? Because the curd is made by bacteria. Okay, the curd is made by bacteria. I hope you know that. Okay, now, if you do not know, it's okay. Okay, if you do not know, it's okay. So, basically, the curd is made by bacteria, that is lactic acid bacteria, okay, which is going to produce lactic acid in the milk. Now, when the milk has become acidic at this point of time, At this point of time, the milk protein which is there, which is known as casein, it is also going to form into different things, which is why curdling happens. Okay, curdling happens. So it is because of the acid thing that this particular bacteria is releasing. Now this particular bacteria is also responsible for what? The formation of vitamin B12. So in the curd we are also getting vitamin B12, which is very important for our body. So lactic acid bacteria is for vitamin B12. You can remember like that. Cool? Next. Enzyme which is useful to remove the oily stains in laundry. In laundry, as in your clothes, for example, like you were having some oily, some chow mein or stuff like that, it fell on your dress and now you have to wash it. So, which enzyme should be definitely there to remove this oily stain? First of all, you should be realizing that oil means it is talking about, you're talking about lipids. Oil is a fat, right? Fats comes under what? Lipids. So basically you should be having something that is going to break down the lipids. Who is the one that is going to break down the lipids, my dear children? It is the lipase or lipase. Lipase is the one that is going to break down the lipids. So this is very important for removing the oily stains in the laundry. Cool? So C becomes the answer for this. Amylase is for carbohydrate, protease is for proteins. Okay, and then some other protein again, some other thing that is again for proteins only, but nothing to do with lipids. So lipase is the one that is the correct answer. Now, the technology of biogas production was developed in India due to the efforts of. So it is given in your NCERT textbook only. Okay, so there were two groups, or basically two very big communities which were responsible for developing biogas production. Now biogas production is really important. This biogas can be used as fuel in the case of kitchen. Okay, food can be made with the help of that, or probably as a fuel for electricity. So it is kind of really important. But who did it? Who was the person? Who were the people behind it? It's a group of people actually who come under this and this particular group. KVIC and IARI. Do you want to know the full form? Full form is right over here. So it is Indian Agricultural Research Institute, my dear children, and also Khadi and Village Industries Commission. What? Indian Agricultural Research Institute. Now the question can be framed in this format also. Please write the full form of AIRI. So you should be knowing Indian Agricultural Research Institute, Indian Agricultural Research Institute, KVIC, what is it? Khadi and Village Industries Commission. Khadi and Village Industries Commission. Okay, so they were the people who were responsible for what were developing biogas production in our country. So I'll say what is the answer for this? Both A and B. So there's one option who's oh my god, yeah, who says that both of them are correct. So that is both A and B right over here. Okay, you remember one, so you ticked one, you will lose your mark. So please, my dear children, there are two group. Okay, so that is this. Next question. Which among the following products of microbes is not obtained from fungi? Penicillin. Penicillin is an antibiotic. Antibiotic as in it is responsible for killing bacteria. Now penicillin is obtained from a fungus itself, that is Penicillium notatum. It is from a fungus. What about statin? Statin is something that lowers the blood cholesterol. You will see a lot of questions related to that. Okay, statin is something that stabilizes the blood cholesterol level in your body, and that is again being released by another fungus which is Monascus purpureus. Monascus. Here it is Penicillium. Penicillium notatum. Now Swiss cheese, I'll keep it for there sometime. Okay. Now cyclosporin A, which is over here, it is again something really important because it acts as immunosuppressive agent. For example, you have some kidney transplant or something like that. So whenever our body sees that this is something foreign that comes inside us, it will react to it, it will fight against it. But we want this kidney. At this point of time, we want our body to be suppressant. Okay, we want our immune system to be suppressed a bit, and for that we have to take immunosuppressive agents. And one such immunosuppressive agent is cyclosporin A, which is again from a fungus that is Trichoderma polysporum. So what we have left? It is Swiss cheese, my dear children. Swiss cheese again, it is obtained from bacteria. It is over here, Swiss cheese has big holes which is done by Propionibacterium shermani. So that is how I remember it. So it is the one that releases too much amount of CO2, which is why there are so many holes in there. So it is done by a bacterium, not by a fungus. So this is the odd one out over here. So I'll say this is your answer. So what is the product which is not obtained from fungi? This is the one that is not obtained from fungi, rest all of them are. Okay, with this information, you get to know about penicillin, statin, cyclosporin, everything. Now this will be given in other questions as well, and you can solve a lot of questions, trust me with that. Okay, chalo. Now 7th question. So this is the exact thing that I have written over here. Penicillin is produced by mold Penicillium notatum. Cyclosporin A is immunosuppressive agent in organ transplant patients. It is produced by fungus Trichoderma polysporum. Now the large holes in Swiss cheese are due to production of large amounts of CO2 by the bacterium named Propionine, Bacterium shermani, and statins produced by yeast. Yeast is what, children? Yeast is again a fungus. Don't say, ma'am, it is produced by yeast, not a fungus. Bacha, yeast is fungus, the unicellular fungus, the only unicellular fungus that is there, it is yeast. So it is produced by yeast. Monascus purpureus have been commercialized as blood cholesterol lowering agents as well. Done? Okay, now this is very, very, very important. It will give you so many questions. So next question. Okay, match the column. Cyclosporin A, you studied right now. Cyclosporin A is responsible for immunosuppressive agent. So A goes with 3. Let's see. A goes with 3. So this and this option, let's cancel it out. Eliminate that. Streptokinase. Now this is something new. I do not know about it. Let's keep it there then. If you do not know, let's keep it there. What about statins? Statins, you know, statin stabilizes the blood cholesterol, right? So I remember like that. It is actually a blood cholesterol lowering agent. So statin stabilizing, it becomes all the more easy. So C is with 4. C is with 4. There's only one option with C is 4. Let's see the other thing. Penicillin. Penicillin is there. Penicillin is what I told you. It's an antibiotic. It kills the bacteria. So it becomes, D becomes with 2. D comes with 2. So B, we are left with what? Clot busters. Streptokinase that you see over here, it is released from a bacteria that is Streptococcus, my dear children. Streptococcus. And Streptococcus is, this particular streptokinase which is there, it is responsible for opening up the clots in our body. So whenever someone has heart attack or some other problems, where basically there is a clot in their blood vessels, to remove that, the first thing that is given is streptokinase, my dear children, because it acts as a clot buster. So over here, this becomes your correct answer. Cool. So one new information over here. Rest of things we studied in the previous slide. Chalo. Next, the large holes in the Swiss cheese are made by Propionibacterium shermani. Okay, and it is releasing a large amount of CO2. Since it is releasing a large amount of CO2, that is why it is. Okay, anyways, so large holes in Swiss cheese are made by a bacterium producing a large amount of CO2, my dear children. A straight question. Why am I teaching you like that? I don't need to do like this and thing. It becomes all the more easy for you to recollect and remember sometimes. Oh, ma'am became cringe and did something like that. Oh, I remember something like that. So it becomes all the more easy for you to relate and learn, which is why I am doing all this action and drama. I hope it works for you. Okay, I hope you are able to understand. My dear children, do let me know if you're not able to understand. Do let me know if you're able to understand. I'll be all the more happy. If I'm not able to make you understand, I'll improve myself. If I'm already good at it, then you have to tell me. Otherwise, how will I be motivated? Yeah? So, yes children, this is the answer for this. Now, let's get into the next question. In sewage treatment, secondary treatment is considered highly significant because in the sewage treatment, the first treatment is known as primary treatment. What happens in primary treatment? Two things: filtration and sedimentation. Filtration as in something that can be filtered very easily. For example, large things, they float around. Okay, so I will remove that. Sedimentation as in some things becomes large and they get precipitated. Now if I remove the precipitate, what will I be left with? I will be left with certain fluid things, which is known as the primary effluent. Rest of the things are being removed. So with the help of sedimentation and filtration, I am removing all the things, and now I have the primary effluent which will go. What children? Primary effluent. Primary effluent which will be now taken for what? Secondary treatment. Cool, that is how it will work. Now, in sewage treatment, secondary treatment is considered highly significant because, yeah. So, we have taken it for secondary treatment. Now, what happens over there? In there, we are going to give so many aerobic bacteria. This aerobic bacteria which is there, my dear children, it is going to utilize oxygen it is going to utilize oxygen to break down organic compounds, to break down the organic compounds which is there. Okay, now I'll tell you one very important thing. You have to be carefully hearing me. Okay, so there is something that is known as BOD, or biochemical oxygen demand. Okay, biochemical oxygen demand, BOD. I always mess it up. It's BOD, yeah. So now, what is BOD? It's the amount of oxygen. It is the amount of oxygen consumed by a bacteria to break down organic matter in 1 liter. In 1 liter of whatever thing has been treated. Okay, so what is it? For example, I am a bacteria. How much amount of oxygen will I require to break down the organic matter? Okay, that is what is BOD. So suppose the organic matter is way too much. That means BOD is also way too much. The amount that I need is also way too much. The organic matter is way too less. That means BOD is also way too less because I need less oxygen. Anyways, it is less. So basically, this happens. So my goal in secondary treatment is to make the BOD's amount really less. That is, I would have to reduce the amount of BOD. When I say I am reducing BOD, that means I am indirectly reducing what, children? Organic matter. When organic matter in the sewage is reduced, that is when the bacteria would need less oxygen, and hence I would be reducing the BOD as well. So in sewage treatment, secondary treatment is considered highly significant because it increases the organic content. No. It helps to remove the debris from the, no. It reduces the BOD level of the sewage. It reduces the BOD level of the sewage. Now suppose the aerobic bacteria is needing more and more oxygen, that means more and more organic matter is there, and when this happens, the aquatic organisms which are there in the water, they will die because they are not getting enough amount of what? Oxygen, my dear children. So that is a problem in the case of aquatic organisms if the sewage is mixed with water. Okay, aquatic life. Now over here we are treating it separately, that's a different thing. So hence, what am I saying? I did filtration and sedimentation in primary treatment. I got the primary effluent now. Now I am going to treat it further with the help of secondary treatment, where I will be treating it with aerobic bacteria first, okay, which will need oxygen to break down the organic compounds. It will need oxygen to break down the organic compounds, my dear children, and hence over here I will say the BOD is really high initially. Later on, when the aerobic bacteria breaks down almost all the organic compounds, the BOD reduces. The BOD will reduce, my dear children, and when this process happens, okay, I am telling you one extra thing. When this process happens, you get to see some flocks. You get to see something which is known as flocks, my dear children. Flocks is a mess. It's a mesh-like thing. It's a mesh-like thing formed of bacteria as well as fungi. Formed of bacteria as well as fungi as well as little proteins and things like that. So, the mesh-like thing that is being formed, it's just bacteria and everything. It's very, very important for the sewage treatment. Those bacteria, those fungi are the ones that is going to treat this particular sewage. Okay. Now, after doing this, what do I do? In the secondary treatment itself, one thing that I do is, again I will separate everything. Okay. Again I will separate everything. Now, the effluent that I am having, I am going to treat it with anaerobic bacteria. The next step is going to be, I am going to treat it with anaerobic bacteria. That is the one that does not need oxygen, my dear children. They do not need oxygen. For example, methanogens. When I do that, what is the use of that? When I do that, what is the use of that? Can you guess what is the use of that? So basically, whatever is left behind, some things might be left behind, I am going to treat it further with the help of anaerobic bacteria. The water which is there, the effluent which is there, I am going to release it to the water, river and things like that, no problem with that. But whatever organic or whatever, let's say, solid thingy is there, I am going to treat it with anaerobic bacteria. Why? Because these anaerobic bacteria are going to produce methane with the help of it. Are going to produce methane with the help of it, and this methane is going to act like what? A biogas, my dear children. So in the next step, I am making the use of the sludge that is I am having, and I am going to treat it with anaerobic bacteria, my dear children, which will be producing methane, which is a biogas, and this biogas can be used in different ways. Okay, and the rest of the water which is being treated can be removed also easily, so that's not a problem. So in that way, we are properly utilizing everything. Okay, so I just explained you that as well, because you will be seeing the questions further. So in sewage treatment, the secondary treatment is considered highly significant because it reduces the BOD level of sewage. Now this is the second thing. It helps in production of biogas. This is the second thing that happens later on. So I won't say this is entirely correct as of now. First thing is first. This is significant. Okay. Next, ruminant animals can digest cellulose in their food, whereas human beings are unable to do so. This is because ruminant animals, as in your cattle and things like that, they are able to digest something that is known as cellulose. Why is it so? Because in their gut, my dear children, in the cattle's gut, you have methanogens. You have methanogens plus other bacteria. Okay, this bacteria will be the one that will be releasing an enzyme that is known as cellulase, which will be acting on what? Cellulose. But for its action, there should not be hydrogen and everything there. So methanogens also play a major role. How? They are going to act on the hydrogen and everything which is being produced while the breakdown of things, such that there is no hydrogen. Okay, such that there is no hydrogen. Instead, the hydrogen, the carbon dioxide, everything together is made to be methane. And now since there is no hydrogen and everything, this bacteria can easily act on cellulase to form cellulose. So I'll tell you once more, this bacteria which brings out cellulase for breakdown of cellulose has just one condition. Please do not put me in this area where there is too much about hydrogen. I won't be able to work really well. So there is a role of methanogens who act on the hydrogen as well as carbon dioxide to produce what, my dear children? Methane. So that problem is sorted. So these guys have to be together. That's when it works properly. So the ruminant animals can digest cellulose in their food, whereas human beings are unable to do so. First thing, we do not have methanogens. Second thing, we do not have other bacteria also which can be producing cellulase. Okay, so methanogens are absent in human gut, methanogens are present in human gut. Cellulose is a complex sugar, cellulose reduces the bulk of the food. It is what methanogens are absent in the human gut. This becomes the answer. Cool? Okay, chalo, next. Identify the incorrect statement with reference to biocontrol agents. What do you mean by biocontrol agents? So, you know children, we use pesticides, chemical things, okay. We used chemical pesticides and all for killing the pest. But this is not really good because these pesticides could seep into the soil and then into the water bodies, and we are going to drink the water bodies, and these chemicals get inside our body. So, instead of that, we are going to use biocontrol agents which are living organisms that could be feeding on pests. For example, you have dragonfly. The dragonfly that you see eats the mosquito larvae. Okay. For example, you might have seen the red and the black beetle that you have having. That beetle which is there, it is ladybird. Okay, the ladybird which is there, the ladybird beetle which is there, it is the one that feeds on aphids. Again, something that causes a huge lot of trouble. Okay, in farming. So basically, I am using animals, or basically I am using organisms to feed on the pest, and they are known as biocontrol agents. So you have to find what the incorrect statement with reference to biocontrol agents. They do not show any negative impact on crop plant. Yes, they do not. They help to increase the use of synthetic pesticides. They do not help, right? So this is a wrong statement. They help to increase the use of synthetic pesticides. No, they do not promote the use of synthetic pesticides. It's an alternative for that only, right? They are significant in treating ecologically sensitive area. It's really good. They do not affect the non-target pests, the pests that do not want to target. They are not going to target them. So this is all true points, but this point that you see is incorrect. Totally incorrect, totally baseless. Cool? Chalo, next question. A farmer has applied chemical fertilizers in his crop field for many successive seasons. In the next season, the crop growth was poor as soil lost its fertility. So, because of biofertilizers and things like that, what happens? Eventually, after a point of time, either there will be too much amount of one type of nutrient present in the soil, such that it could make the other nutrients devoid in the soil. So, this has a huge, it can lead to soil erosion, lot of things. So, eventually, because of these chemical fertilizers which are used, the soil will lose its fertility. So what can be done? Suggest a suitable microorganism that replenishes the fertility of soil in this field. So over here we can use something that is known as biofertilizers. And what do you exactly mean by biofertilizers? Biofertilizers are the one that is going to help in fertilization, not fertilization, basically for increasing the nutrient capacity of the soil. How? For example, I have a bacteria who fixes nitrogen, who takes in the atmospheric nitrogen and converts into some other form, nitrates and nitrites, and give it to the plant. So, don't you think it is increasing the soil fertility? Yes, it is. So, similarly, you have so many different type of organisms, my dear children, Annabana, this, that. So, over here you have nostoc, which is something which is a cyanobacteria, my dear children, and it is responsible for, again, was say fixing nitrogen over there, for increasing the amount of nitrogen in the soil. So nostoc is the one that plays a major role over here. Cool? Next. The microorganisms involved in flock formation during sewage treatment are, I told you, flock is being made with the help of what? Aerobic bacteria and fungus, my dear children, and also the protein filaments and everything that is being left behind. So it is not anaerobic bacteria, it is aerobic bacteria and fungus. Anaerobic treatment is done later on. First aerobic treatment is done. Okay, so anaerobic is not the correct answer, aerobic is the one. So aerobic bacteria and fungus forms the flock which is there. Okay, I told you question will be coming. Now, match the following bacteria of column 1 with their commercial products of column 2. So, column 1 has lactobacillus. Lactobacillus is a lactic acid bacteria, my dear children. So, it is a lactic acid bacteria, so that means it is going to help in curd formation. So, lactobacillus is lactic acid bacteria. A goes with R. A goes with R. I can remove one option. Okay. Aspergillus niger. Now, this is something that you have not studied as of now, at least not in this particular session. So, Aspergillus niger, it is a fungus and it is responsible for the production of citric acid. Okay, Aspergillus niger is the one that is responsible for production of citric acid. So, B goes with S. So, B goes with S. So, this and this are the options. Now, we cut this. Okay. Now, Acetobacter aceti. The name suggests what it produces, Acetobacter aceti. It is going to produce what? Acetic acid. C with Q. C is going to go with Q. So, this becomes the one. And can I just remove this option? Yes. What is Clostridium butylicum? It is going to produce butyric acid. The names are all very easy to remember for you guys. As in, I am not saying the names are easy. I am saying the function that they do. For example, butylicum, butyric acid, Acetobacter aceti, acetic acid. Aspergillus niger has a little different thing. So, the citric acid is going to produce citric acid. So, over here, Aspergillus niger is the only fungus. This is a bacteria. This is the bacteria. This is also a bacteria, my dear children. You will be seeing so many questions around this as well. Okay? You will be seeing so many questions around this as well. So, Aspergillus niger, citric acid, Acetobacter aceti, acetic acid, Clostridium butylicum, it is butyric acid. So, this becomes your answer. Okay? Sure. Next, which one of the following has been commercialized as a blood cholesterol lowering agent? Tell me the one that stabilizes the blood cholesterol level. How do I remember it? Statin, stabilizing. Easy. So, I don't have to see other options as well. Direct hit to that particular option. Next question. As the organic matter increases in water body, the BOD, biochemical oxygen demand. Organic matter is increasing. I am a bacteria. I would need more oxygen for the breakdown of this. So, obviously, needing more, consuming more oxygen means I am increasing. Oxygen demand is increasing. So, biochemical oxygen demand increases. As the organic matter increases in the water body, the BOD also increases. Okay? Chalo. If the question was, as the organic matter decreases in water body, the BOD will decrease. Does not need much oxygen, so it will decrease. Now, the puffed up appearance of dough is due to fermentation by bacteria. Identify the gas liberated during the process. The puffed up appearance of dough is due to fermentation by bacteria. Identify the gas liberated during this process. So, my dear children, whenever fermentation is happening, there is a release of a gas which is known as carbon dioxide. So, it is because of that gas that there is a puffed up appearance. Nothing else. It will be always carbon dioxide. Please remember that. Fermentation, carbon dioxide. Fermentation, carbon dioxide. Okay. Next question. The primary treatment of sewage water involves sludge digestion, aerobic bacteria activity, anaerobic bacterial activity, filtration and sedimentation. I told you the primary treatment is basically when you filter out the things that are floating around and also the things that are sedimenting around. So, it is filtration and sedimentation. Further, you treat it with aerobic bacteria, then anaerobic bacteria, and then it's done. Okay? Chalo. A person was admitted to the hospital as he had myocardial infarction. A cardiologist injected him with streptokinase. Why? Streptokinase. I told you streptokinase is a clot buster, my dear children. It is going to remove the clot which is there. So, obviously, this is the answer. It is not the one that reduces hypertension or reduces the blood cholesterol level or stimulates the heartbeat. No, my dear children, it is just a clot buster. And who releases it? It is again a bacteria whose name is Streptococcus. Cool? Yes. Now 20th. BOD refers to the oxygen required for bacteria to grow in 1 liter of effluent. The amount of oxygen consumed if all the organic matter in 1000 milliliter of water were oxidized by bacteria. The amount of oxygen, I am repeating again, amount of oxygen consumed if all the organic matter in 1000 milliliter of water were oxidized by bacteria. This is what is the answer. The amount of oxygen released? No. The amount of oxygen released? No. It is the use consumed, okay, how much amount of oxygen is being consumed by a bacteria, okay, not by a bacteria, it's basically the amount of oxygen consumed by the bacteria to oxidize what organic matter present in what? 1 liter of water, or 1 liter of water can be said as 1000 milliliter of water. So this becomes your answer. Consumed, BOD means you are consuming. Oxygen demand, as in someone is consuming, that is why demand is high, right? Okay. Now Ernst Chain and Howard Florey's contribution was establishing the potential of penicillin as an effective antibiotic, discovery of streptokinase, production of genetically engineered insulin, discovery of DNA sequence. My dear children, okay, so penicillin was released, penicillin was found out accidentally. So what was happening? Alexander Fleming, okay, he was doing his experiment when he found that there was one unwashed petri dish present, and this unwashed petri dish was not allowing there was something in there in this unwashed petri dish that was not allowing the growth of Staphylococcus. There is another bacteria. He was trying to grow a bacteria up there, okay, and he found that in this particular unwashed petri dish, this particular bacteria was not growing. So then he found that there was a mold of penicillin. There was a mold of Penicillium notatum, my dear children, which was releasing penicillin probably, which is why the Staphylococcus bacteria which is there, it is not able to survive. Okay, but then it became like a big thing. The actual potential of it was recognized later by Ernst Chain and Howard Florey, which is why 3 of them, Fleming, Ernst Chain as well as Howard Florey, received a Nobel Prize as well for this. So it was, their role was in establishing the potential of penicillin as an effective antibiotic. Okay? Yes. Now during sewage treatment, biogas produced includes what? So what is that the biogas will be containing? First thing, obviously there should be methane. You cannot exclude methane, okay. Methane and carbon dioxide is a short thing, and you know it smells really bad. So it has to be hydrogen sulfide as well. So what do we have, my dear children? Methane, hydrogen sulfide and carbon dioxide. Methane also smells bad, but yeah. So hydrogen sulfide, nitrogen, methane, no. Nitrogen is not there. Methane, oxygen, no there. Hydrogen sulfide, methane, sulfur dioxide, no. So it will be methane, hydrogen sulfide, H2S, as well as what? Carbon dioxide. This becomes your answer. What is there in the biogas? This is there in the biogas. Next, floc is what? I told you it's a mesh-like thing that is formed by association of bacteria and fungal filaments in sewage treatment. The primary cells produced in sewage treatment, the effluent in primary treatment tank obtained during sewage treatment, the type of biofortified food, oh my god, too much complicated. This is the answer. It's what? It's a mesh-like structure formed by what? The association of bacteria and fungal filaments. What bacteria, my dear children? Aerobic bacteria, to be very specific. It is what? Aerobic bacteria which is there. Cool. Next, Roquefort cheese is ripened by using what? Bacterium, type of yeast, cyanobacteria, fungus. Again, the same question in different format. Bacterium, no. Type of yeast, no. Cyanobacteria, no. It's a fungus. Yeast is also fungus, but I told you it's not a type of yeast that was being used. It was Penicillium, some other type of Penicillium. Forgot the name. It is not notatum. Some other type of Penicillium which was responsible for producing something. Basically, it's a fungus only. Penicillium is a fungus, you know, right? Now, that is responsible for ripening of what? Roquefort cheese. Next question. Match the microbial products listed under column 1 with the related microbes given under column 2. Okay, choose the appropriate option for the given choices. Now it will become easy for you because you have studied a lot of things, right? Citric acid, Aspergillus niger. A with R, you have 2 options like that, so I'll cut these 2 options, eliminate. Cyclosporin A, cyclosporin A is produced by another fungus, that is what? Trichoderma polysporum. I already told you what is the role. It is an immunosuppressive agent. So B with S, both are hand in hand. Statin is the one that is responsible for stabilizing, and it is produced by Monascus purpureus. Monascus purpureus. So C is with Q. Okay. C is with Q. Now you have just one option that is different. Gobar gas. Gobar gas has what? Methanogens. Okay, gobar gas is being produced with the help of what? Methanogens, my dear children. Methane, so you can relate. So it is D goes with P. So this becomes your answer. Whereas you can eliminate this option. Okay. Next question. Which of the following is correctly matched for the product produced by them? Okay. Methanobacteria, lactic acid. No, methane. It produces methane. Penicillium notatum, acetic acid. Saccharomyces cerevisiae, ethanol. What is Saccharomyces cerevisiae, my dear children? Saccharomyces cerevisiae is the yeast. And yeast during fermentation. Yeast, when it undergoes fermentation, it produces CO2, it produces ethanol and a little amount of ATP also. So ethanol is being produced by this yeast. Okay, ethanol. Acetobacter aceti, antibiotics. No. So this is the correct answer. Okay. So that is something unique that has come. This is a question that I took from NEET. Okay, there are 5 questions that I took from NEET category as well. Now, which one of the following can be used as a biocontrol agent in the treatment of plant disease? Any idea? Which can be used as a biocontrol agent? I told you biocontrol agent as in a biological organism which is going to control the pests, right? So there is, pest can be anything. It can be a fungus, it can be a bacteria, it can be another bacteria, it can be anything. Or an organism, insect also. So there is trichoderma. Okay, trichoderma is a fungus itself, but it is also responsible for killing other fungus. It is also responsible for killing other fungus, which is a pest. So these fungus are present in the roots of certain plants, and trichoderma eventually eliminates them. So it is acting as a biocontrol agent. Okay, not chlorella, not anabana. Anabana is chlorella. Is for something else. Okay, anabana is for basically, what do you, okay, sorry. Anabana fixes nitrogen, lactobacillus produces lactic acid and things like that. For trichoderma, you have this. It's a fungus itself. Trichoderma polysporum you have studied, right? It produces cyclosporin A as well. So trichoderma, which is there, it's a fungus and it kills other fungus which is acting as a pest in the roots of the plant. So it is going to act like a biocontrol agent. Next thing, microbe used for biocontrol of pest butterfly caterpillars is butterfly caterpillars. So that means you're talking about insect thingy. Insects can be eaten up by other insects, but usually I'm telling you the caterpillars, the larvae and everything. Insect larvae are being damaged with the help of something which is known as Bacillus thuringiensis. Okay, it is done by Bacillus thuringiensis. My dear children, what is it? It's a bacteria. And this bacteria produces an inactive toxin in itself. It produces inactive toxin. Now, when this particular caterpillar is going to eat this particular bacteria, what happens inside the caterpillar's gut? Inside the gut, this particular inactive toxin is going to become active and going to damage the cellular layer up over here. It is going to cause immense damage to the layer up, epithelial tissues up over here, which is eventually going to kill this caterpillar. Okay, so Bacillus thuringiensis is going to produce inactive toxin, which when inside the body of the caterpillar or an organism becomes active and later kills it completely. Okay, so Bacillus thuringiensis is the answer for this. And some plants, okay, we put the genes which is responsible for producing the inactive toxin over here. This gene which is going to produce the toxin, we are going to put it in some plants. So the plants itself can produce the toxin. It does not depend on the bacteria. You call them as BT plants. So for example, BT cotton, BT brinjal and etc. etc. Okay, so microbes used for biocontrol of pest butterfly caterpillar is Bacillus thuringiensis, my dear children. So this becomes the answer. Last 2 questions and then we are done for it. Which one of the following is not a biofertilizer? Biofertilizer, an organism that will increase the nutrient quality of the soil, right? Agrobacterium, Rhizobium, Nostoc, Mycorrhiza. So Rhizobium, Nostoc, Mycorrhiza, all of them, they are going to increase the soil fertility. Agrobacterium, which is there, it is basically a bacteria. Agrobacterium tumefaciens is the name, and it's a pest actually, not a pest, but it is the one that causes huge lot of trouble for plants. Okay, it is the one that causes huge lot of trouble for the plants, but we are going to use it in a positive way. How does it cause trouble? It causes cancer in plants. Okay, it has a plasmid which is known as TI plasmid, tumor inducing plasmid, which has a DNA, transfer DNA. Transfer DNA, it produces protein which causes cancer and stuff like that. What do we do is that we remove that transfer DNA part and we put our thing, whatever we need to insert into the plant, and we give it off. Anyway, this guy will go and infect, but this guy does not know that it does not have the transfer DNA. Instead, it has something else that we have inserted. So it is usually acting as a vector, but this is the one that usually causes trouble. Only if it is genetically modified. Only if it is genetically modified, that's when it acts like a vector, my dear children. So which one of the following is not a biofertilizer? It is Agrobacterium, which is not a biofertilizer. Cool. The last question that we have. Select the correct statement for the from the following. Biogas is produced by the activity of aerobic bacteria on animal waste. It is done by anaerobic bacteria. You have to find the correct one, right? So this is done by anaerobic. Anaerobic should be the one. Methanobacterium is an aerobic bacterium. It's an anaerobic bacterium. Biogas, commonly called gobar gas, is pure methane. I told you biogas has hydrogen sulfide, carbon dioxide, methane. So it is not pure methane. So what is left? What could be correct? This could be the correct one. Activated sludge sediment is in settlement tanks of sewage treatment plant is a rich source of aerobic bacteria. Activated sludge sediment. Okay. So after doing the secondary treatment, in the secondary treatment when you treat with aerobic bacteria, okay, then you get an activated sludge which is there. You get a sludge which is there. This has enough amount of what? Aerobic bacteria, my dear children. And this little part of activated sludge is being put again in some other treatment plants where treatment is required, because it is a rich source of aerobic bacteria, right? So this becomes the answer for that as well. So activated sludge sediment in settlement tanks or sewage treatment plant is a rich source of aerobic bacteria. This is entirely correct, and with this we get 30 on 30, my dear little champions. So do let me know your scores as well once you're done with all the questions. Okay, see for how much are you getting. It's okay even if you do not get, you are studying, you're still studying. Okay, if you are trying, and that is what matters the most. Your efforts matters the most. So keep on trying, do not get demotivated, my dear children. You will be acing the exam, I'm pretty sure of that. Just give your 100%. Yeah, and if you like this particular video, do not forget to subscribe to our little channel, that is Deeksha, Karnataka, my dear children. And also do let me know if you have any doubts. I will be clearing all of it for you guys. That's all. Bye-bye. Take care.